Welcome to the deep dive. Today, we're embarking on a really fascinating exploration, a journey deep into the incredible machinery that powers literally every move you make. From, blinking your eye to that tireless beat of your heart. We're taking a deep dive into muscle tissue, and we're pulling all our insights from a fantastic resource, Histology, a text and atlas, 8th edition. Our mission today is to give you a really clear understanding of the microscopic world of muscle. We'll unpack its intricate anatomy, the tiny cellular bits that make it work, the different types of muscle you find all through your body, and importantly, those connections to real-world health issues. Think of this as your shortcut to understanding the amazing biological engineering behind all movement, whether you control it consciously or it's just humming along in the background.
And it really is amazing. We're going to journey through the intricate details of skeletal muscle, that's the stuff that lets you run and jump. Then there's cardiac muscle, the unique tissue in your heart that, well, never rests. And smooth muscle, too, the sort of silent worker in your internal organs. We'll highlight their unique structures, how they actually manage to contract, their really remarkable roles, and even touch on how they repair themselves, which is sometimes quite limited. Get ready for some serious aha moments as we connect these tiny, invisible details to your everyday life and health.
So how does all this incredible machinery actually work? Let's start with the the big picture, the core purpose of all muscle.
Movement.
Right, movement. But it's not just lifting weights or running, is it?
No, exactly. It's also about those tiny involuntary actions inside your body that keep you alive. Things circulating blood, digesting food, that kind of thing. Fundamentally, muscle tissue is built from these specialized long cells, all lined up neatly, parallel to each other, and they're primarily designed to do one thing: contract. This ability comes down to two key molecular players, tiny filaments inside the cells. You've got the thin filaments, mostly made of a protein called actin, and the thick filaments, mainly made of myosin. And the cell's internal fluid, where all this action happens, we call that sarcoplasm.
Okay, so when you first look at muscle under a microscope, what's the most basic way we categorize it? What are those first visual clues?
Well, the first big split is whether the muscle cells look striated, meaning they have these distinct cross stripes you can see, or smooth, where they just, well, don't have those stripes. It's a really obvious difference under the scope.
Striated versus smooth.
Oh.
Okay.
And then, within striated muscle, we break it down further. First, there's skeletal muscle. This is the type attached to your bones, responsible for moving your skeleton around, maintaining posture, even those incredibly precise eye movements. Then there's a subtype, visceral striated muscle. Looks identical, but it's found in soft tissues like your tongue, your pharynx, parts of the diaphragm, and esophagus. Super important for speech, breathing, swallowing. And finally, distinct from those is cardiac muscle, found only in the heart wall and the large veins right where they empty into the heart.
So those visible stripes, the cross striations we see in skeletal and cardiac muscle, they're not just random patterns.
Not at all. They're actually the result of a very specific, highly ordered arrangement of those thin and thick myofilaments we mentioned. Almost like a microscopic crystal lattice. Smooth muscle just lacks that high degree of internal order, so no stripes. It looks smooth.
Makes sense. Okay, let's jump into skeletal muscle then, the voluntary powerhouse. Each skeletal muscle cell, or fiber as it's often called, is pretty unique, isn't it? It's not just a single cell in the typical sense.
That's right. It's actually a syncytium. During development, many individual muscle precursor cells, called myoblasts, fuse together. This creates one very long, large cell that contains multiple nuclei.
And where are those nuclei usually found? That's a key feature, right?
Absolutely. They get pushed to the periphery, right underneath the cell membrane, the sarcolemma. Seeing those nuclei lined up along the edge is a dead giveaway for skeletal muscle fiber. The histology text shows this really clearly in figure 11.1b in plate 21.
And these fibers aren't just loose. There's a whole structure holding them together.
Oh, definitely. They're organized by layers of connective tissue. Think of it as internal scaffolding and packaging. You have the endomysium, a very delicate layer wrapping around each individual fiber. Then fibers are bundled together into groups called fascicles, and each fascicle is wrapped by a thicker layer, the perimysium. And finally, the whole muscle itself is encased in a dense sheath called the epimysium.
And these layers aren't just for structure.
No, they're crucial. They transmit the force generated by the contracting fibers to the tendons and bones. Plus, they carry the blood vessels and nerves that the muscle needs to function. It's all integrated.
Now, what about different types of skeletal muscle fibers? I understand they're not all the same.
Correct. They specialize. We mainly classify them based on how fast they contract, how they make energy, and how quickly they fatigue. There are broadly three types.
Okay. Break those down for us.
First, you have type I fibers, often called slow oxidative. These are typically smaller, darker red because they're packed with mitochondria, the energy factories, and myoglobin, which stores oxygen. They contract relatively slowly, but are incredibly resistant to fatigue. Think endurance muscles.
Like postural muscles in your back, or what a marathon runner relies on.
Exactly. Perfect examples. Then you have type II, the fast glycolytic fibers. These are on the other end of the spectrum. They're larger, appear lighter or whitish because they have less myoglobin and fewer mitochondria. They rely heavily on anaerobic glycolysis, quick energy production without oxygen. They contract very quickly and powerfully, but they fatigue fast. Think sprinters or the muscles making rapid eye movements.
Got it. Quick bursts of power. And the middle ground.
That would be type dia. The fast oxidative glycolytic fibers are sort of intermediate. Medium size, lots of mitochondria and myoglobin like type I, but also capable of fast contraction and significant anaerobic activity like type I. They're fast twitch, but also reasonably fatigue resistant. Good all-rounders for sustained power.
And that mention of myoglobin in type I fibers, there's a clinical connection there, right? If muscle gets badly damaged.
Yes, absolutely. In severe crush injuries or other major muscle trauma, a condition called rhabdomyolysis, large amounts of myoglobin can be released from the damaged fibers into the bloodstream. This can actually overwhelm the kidneys as they try to filter it out, potentially leading to kidney failure. So detecting myoglobin in the urine or blood is a really important indicator of serious muscle injury.
Wow. Okay, let's zoom in even further now. Inside these fibers, what's actually doing the contracting work?
That would be the myofibrils. So these are the fundamental structural and functional subunits within the muscle fiber. They're essentially bundles of those thin and thick myofilaments we keep mentioning. And it's the highly organized repeating arrangement of these myofilaments within the myofibril that creates those characteristic cross striations.
The stripes.
Exactly. You see alternating dark bands called A bands and light bands called I bands. The basic contractile unit is the sarcomere. It's the segment of the myofibril between two successive Z lines which run down the middle of the I bands.
So the sarcomere is the engine.
It's the fundamental engine, yes. Contraction is essentially the shortening of thousands of these sarcomeres all at once. Within the sarcomere, you also see the H band, a lighter region in the middle of the A band where only thick filaments are present, and the M line right in the center, which helps hold the thick filaments together. Figures 11.4, 11.5 in the text show this beautifully.
Okay, let's talk about those filaments again. The thin filaments, mostly actin, you said.
Primarily F-actin, which forms a double helix structure. But crucially, it has regulatory proteins associated with it. There's tropomyosin, which in a relaxed state physically blocks the sites on actin where the myosin heads want to bind. And then there's the troponin complex, which is the key calcium sensor. It has three parts. TNC binds calcium ions, TNT binds to tropomyosin, and TNI inhibits the actin-myosin interaction.
So troponin is like the switch that calcium flips.
Exactly. Calcium binding to TNC causes a conformational change that shifts tropomyosin out of the way, exposing the myosin binding sites on actin. Contraction can then proceed.
And the thick filaments.
Those are composed mainly of myosin the second molecules, typically 200, 300 per filament. Each myosin molecule has two globular heads. These are the business end, containing sites that bind both actin and ATP, the energy currency. Then there's a long tail. These molecules assemble tail to tail, forming a bipolar thick filament with the head sticking outwards, except for a central bare zone where there are only tails. Figure 11.8 and 11.9 illustrate this structure.
But it's not just actin and myosin floating around, right? There must be something holding this precise structure together.
Absolutely. There's a whole cast of essential accessory proteins. Think of them as the molecular scaffolding and stabilizers. Figure 11.10 shows this well. You have titin, a huge elastic protein that anchors the thick filaments to the Z line and acts like a spring, preventing overstretching and helping the sarcomere recoil. Alpha-actinin is key for anchoring the thin filaments to the Z line. Titin helps link adjacent sarcomeres together. Proteins of the M line hold the thick filaments in register.
And one protein that has huge clinical significance is dystrophin, right?
Yes, dystrophin. It's a massive protein that links the outermost actin filaments to a complex of proteins in the sarcolemma, effectively connecting the contractile apparatus inside the cell to the supportive external lamina outside the cell. See figure F11.2.1 in the text.
And when dystrophin is missing or non-functional.
That leads to Duchenne muscular dystrophy, DMD. It's a devastating X-linked recessive disorder, meaning it primarily affects boys. Without functional dystrophin, the muscle fibers are easily damaged during contraction, leading to progressive muscle degeneration, weakness, and eventual death. It really highlights the critical importance of this single protein in maintaining muscle integrity. There's a lot of research ongoing into gene therapy and stem cell approaches, which offers hope for the future. Folder 11.2 in the text discusses this.
So we have the structure. How does it actually produce movement? The sliding filament model.
Precisely. It's a beautifully simple concept, really. During contraction, the thin filaments slide past the thick filaments. This increases the amount of overlap between them, pulling the Z-lines closer together and shortening the entire sarcomere.
Crucially, the filaments themselves don't actually shorten.
No, they maintain their length. They just slide relative to each other. Think of it like interlocking fingers. Your fingers don't change length, but your hands get closer together. Figure 11.6 shows this process.
And what drives the sliding, that molecular dance you mentioned earlier?
Yes, the actin-myosin cross-bridge cycle. It's a cycle of binding, pulling, and releasing, powered by ATP, detailed in figure 11.11. It starts with the myosin head tightly bound to act in the rigor state. Think rigor mortis, where lack of ATP locks this state.
Okay.
Then, an ATP molecule binds to the myosin head, causing it to detach from the actin. Next, the myosin head hydrolyzes the ATP, breaks it down to ADP and phosphate, which cocks the head into a high-energy, ready position. The myosin head then weakly binds to a new site further along the actin filament. It then releases the phosphate, which triggers the power stroke. The myosin head pivots forcefully, pulling the thin filament towards the center of the sarcomere. ADP is released during this stroke.
And then it binds tightly again.
Exactly. It reattaches firmly to the actin in this new position, ready for another ATP molecule to bind and start the cycle all over again, as long as calcium and ATP are present.
So calcium is the trigger, ATP is the fuel. How is this whole process switched on and off so precisely? You mentioned the sarcoplasmic reticulum, SR, and
Correct.
T-tubules.
Regulation is key. The SR is the cell's internal calcium store, forming specialized sacs called terminal cisternae right next to the T-tubules. The T-tubules are invaginations, little tunnels, of the cell's outer membrane, sarcolemma, that dive deep into the fiber, typically at the junction between the A and I bands and skeletal muscle. Together, one T-tubule flanked by two terminal cisternae forms a structure called a triad. See figure 11.12.
A triad. Okay.
Now, when a nerve impulse arrives at the muscle surface, it travels down the T-tubule. Embedded in the T-tubule membrane are voltage sensor proteins, DHSRs. The electrical signal activates these sensors. These sensors are physically linked to calcium release channels, called RIR1 in skeletal muscle, on the membrane of the adjacent SR cisternae. Activation of the sensors mechanically opens the RIR1 channels, causing a massive flood of calcium ions from the SR out into the sarcoplasm where the myofibrils are.
And that calcium finds troponin C.
Exactly. Calcium binds TNC, tropomyosin moves, myosin binds actin, and the cross-bridge cycle begins contraction.
And relaxation.
Relaxation occurs when the nerve signal stops. Calcium is actively pumped back into the SR by powerful C2 plus ATPase pumps, lowering the calcium concentration in the sarcoplasm. Tropomyosin then slides back to block the myosin binding sites, and the muscle relaxes.
This all starts with a nerve signal, right? At the neuromuscular junction.
Yes, skeletal muscle contraction is initiated by signals from motor neurons. The point where the nerve terminal meets the muscle fiber is the neuromuscular junction, or motor end plate. Figure 11.33, 11.4. The axon terminal of the motor neuron releases a neurotransmitter, acetylcholine, ACEC, into the synaptic cleft, the tiny gap between nerve and muscle. AECH diffuses across the cleft and binds to specific nicotinic AECH receptors on the muscle fiber's sarcolemma.
And that binding triggers the electrical signal in the muscle.
Precisely. It causes depolarization of the sarcolemma, which then spreads down the T-tubules to initiate calcium release. Critically, an enzyme called acetylcholinesterase, ACE, is present in the cleft and rapidly breaks down ADC. This ensures the signal is brief and allows the muscle to relax quickly, ready for the next signal.
And problems at this junction are behind some serious conditions.
Absolutely. Myasthenia gravis, for example, is an autoimmune disease where the body mistakenly produces antibodies that block or destroy those ACV receptors. This impairs neuromuscular transmission, leading to fluctuating and often severe muscle weakness. Folder 11.3 discusses this. Also, certain toxins work here. Botulinum toxin, for instance, prevents the release of AC from the nerve terminal, causing paralysis. Conversely, things like curare, a plant toxin, block the AC receptors themselves.
You mentioned motor neurons. What's a motor unit?
A motor unit is simply one motor neuron and all the skeletal muscle fibers it innervates or controls. The size of the motor unit varies greatly depending on the muscle's function.
How so?
While muscles requiring very fine, precise control, like those moving your eyes, have very small motor units. One neuron might control only a handful of fibers. But large, powerful muscles used for posture or locomotion, like your glutes or back muscles, have huge motor units, where one neuron might control hundreds or even thousands of fibers. This allows for graded force production. Activating more motor units generates more force.
And the nervous system also gets feedback from the muscle, right? It's not just sending commands.
Definitely. There's crucial sensory feedback. Specialized receptors called muscle spindles are embedded within the muscle itself. They detect changes in muscle length and the rate of change, providing information about stretch, figure 11.9 sets. And Golgi tendon organs, located in the tendons, monitor muscle tension or force. This sensory information constantly feeds back to the central nervous system, allowing for smooth, coordinated movement and adjustments to posture.
What about how skeletal muscle develops and repairs itself?
Muscle fibers originally developed from the fusion of precursor cells called myoblasts. This process is tightly regulated by various factors. Now, mature skeletal muscle has a very limited ability to regenerate after injury. However, there are quiescent stem cells called satellite cells located just outside the muscle fiber membrane, but under the external lamina.
The resident stem cells.
Exactly. If a muscle fiber is injured, but its surrounding external lamina remains intact, these satellite cells can become activated. They proliferate, differentiate into new myoblasts, fuse together, and can actually form new functional muscle fiber segments to repair the damage.
But if the damage is too severe,
If the external lamina is disrupted or the damage is extensive, fibroblasts often move in and repair the area with connective scar tissue instead. So the regenerative capacity is present, but quite limited, especially compared to some other tissues.
okay, fascinating stuff on skeletal muscle. Let's switch gears now to cardiac muscle, the heart's tireless engine. Still striated, but different.
Yes, still striated, meaning the basic actin-myosin arrangement in sarcomeres is similar, but there are key differences in cell structure and function. Plate 24 shows cardiac muscle. Cardiac muscle cells, or cardiomyocytes, are much shorter than skeletal fibers, often branched, and they interconnect to form a complex network. And a major distinguishing feature, they typically have only one nucleus, maybe two, and it's located centrally within the cell, not peripherally like in skeletal muscle, figure 11.19.
That central nucleus is a big visual clue then.
Huge clue. Also, the area around the nucleus is packed with mitochondria reflecting the heart's massive energy demands, plus glycogen granules, and, especially in the atrial cells, granules containing hormones like ANF and BNF that help regulate blood pressure and fluid balance.
But maybe the most unique feature of cardiac muscle are those intercalated discs. They look like dark lines crossing the fibers.
Absolutely iconic and functionally critical. Intercalated discs are specialized, complex junctions that connect individual cardiac muscle cells end to end. Figure 11.19, 11.21. They're not just simple connections. They have different components doing different jobs.
Okay, what's in them?
They have a transverse component running across the fiber, which contains fascia adherens. This is the major part, strongly anchoring the cells together and serving as the attachment site for the thin filaments of the terminal sarcomeres. Think of it like molecular Velcro holding the contractile machinery end to end. Also in the transverse part are maculae adherentes, or desmosomes, which are like spot welds, preventing the cells from pulling apart during contraction.
And the other component.
Then there's a lateral component running parallel to the fibers. This is where you find the gap junctions. Gap junctions are crucial. They form channels that allow ions and small molecules to pass directly from the cytoplasm of one cell to the next.
Ah, so that's how the electrical signal spreads so quickly through the heart?
Precisely. This ionic coupling allows the entire network of cardiac muscle cells to depolarize and contract almost simultaneously, functioning as a single coordinated unit of functional syncytium.
What about the calcium handling? Is the SR&T tubule system the same as in skeletal muscle?
Similar concept, but different arrangement. The SR in cardiac muscle is less extensive and less organized than in skeletal muscle, and the T tubules are wider and penetrate the cell at the level of the Z lines, not the AI junction. Instead of forming triads, they typically associate with just one terminal cisterna of the SR, forming a dyad, figure 11.20.
Does this difference affect how contraction starts?
It does, significantly. In cardiac muscle, contraction initiation relies on calcium-triggered calcium release, figure 11.22. The depolarization wave traveling down the T-tubule opens voltage-gated calcium channels, DHSRs, but functioning as channels here, allowing a small amount of calcium to enter the cell from the extracellular fluid. This initial influx of calcium is the crucial trigger, It binds to and opens the RY2 calcium release channels on the SR membrane.
So external calcium triggers internal release.
Exactly. This triggers a much larger release of calcium from the SR stores into the sarcoplasm, which then binds to troponin C and initiates contraction, similar to skeletal muscle. But that initial external calcium trigger is essential for cardiac muscle.
And that RYR2 channel mutations there can cause problems.
Yes, mutations in the gene for RYR2 are linked to certain types of inherited cardiac arrhythmias, like catecholaminergic polymorphic ventricular tachycardia, which can cause fainting or even sudden death, especially during exercise or stress. It again highlights that connection between molecular detail and serious health outcomes.
Does the heart have specialized cells for conducting the electrical signal?
It does. These are called Purkinje fibers, plate 25. They are modified cardiac muscle cells, larger than regular cardiomyocytes, and specialized for rapid conduction, not forceful contraction. They form the heart's intrinsic conducting system, originating the heartbeat and spreading the impulse quickly throughout the ventricles to ensure coordinated pumping. Morphologically, their myofibrils are often pushed to the periphery, and they have a pale, glycogen-rich cytoplasm.
So the heart generates its own beat, but the nervous system can influence it.
Correct. The autonomic nervous system, both sympathetic, fight or flight, and parasympathetic, rest and digest nerves, heavily innervates the heart. But they don't initiate the beat. They modulate the intrinsic rate set by the heart's pacemaker cells, speeding it up or slowing it down as needed.
And what happens when cardiac muscle is injured, like in a heart attack?
A heart attack, or myocardial infarction, occurs when blood supply to a part of the heart muscle is blocked, causing those cells to die due to lack of oxygen. Unfortunately, adult cardiac muscle has very, very limited regenerative capacity. Dead cardiomyocytes are primarily replaced by non-contractile fibrous scar tissue.
Which means that part of the heart loses function permanently.
Largely, yes. This scarring can impair the heart's ability to pump effectively. This is why detecting heart muscle damage early is critical. Proteins specifically found in cardiac muscle, like cardiac troponins, PNI and TNT, leak out of damaged cells into the blood. Measuring levels of these troponins is now the gold standard for diagnosing an MI.
Any hope for regeneration?
Well, for a long time, the dogma was zero regeneration, but more recent research suggests there might be a very small population of cardiac stem or progenitor cells, and some minimal cardiomyocyte division might occur. It's an active area of research, offering some hope for future therapies, but currently, repair is mainly via scarring.
Okay, one more type to cover. Smooth muscle. The involuntary regulator. Visually quite different, right? No striations.
Exactly. Smooth muscle typically forms bundles or sheets of elongated, spindle-shaped fusiform cells with tapered ends. Figure 11.23, plate 26. And the defining feature is the complete absence of those visible cross striations. This immediately tells us the internal arrangement of actin and myosin filaments is fundamentally different from striated muscle.
Tell us about these cells.
Smooth muscle cells can vary a lot in length, depending on their location. They usually have a single, centrally located nucleus. An interesting artifact you often see in tissue sections is that if the cell contracts during fixation, the nucleus can get squished into a corkscrew shape, which is actually a helpful identification feature, figure 11.23. Like cardiac muscle, smooth muscle cells are often connected by gap junctions, allowing them to communicate and contract in a coordinated fashion.
What about their contractile machinery? Still actin and myosin?
Yes, but with key differences. The thin filaments contain actin and a smooth, muscle-specific isoform of tropomyosin. But importantly, they also have two other regulatory proteins, caldasemin and calponin, which help regulate the interaction with myosin. And critically, smooth, muscle-thin filaments do not contain troponin.
No troponin. That's a major difference in regulation.
Huge difference. The thick filaments are also unique. They're made of a specific smooth muscle myosin, SMM, and have what's called a side polar arrangement, figure 11.26. This means the myosin heads project along the entire length of the filament except maybe at the very ends, allowing for interaction with actin over a much greater distance compared to the bipolar filaments of striated muscle. This contributes to smooth muscle's ability to generate strong, sustained contractions.
And instead of Z-lines, smooth muscle has something else.
Right. It has dense bodies, figured 11.24, 11.25. These are electron-dense areas in the cytoplasm and also attached to the inner surface of the cell membrane. They contain alpha-actinin, similar to Z-lines, and serve as anchor points for both the thin filaments and intermediate filaments, like desmin and aventin. Functionally, they are analogous to Z-lines, transmitting the contractile force generated by the sliding filaments throughout the cell and to the surrounding tissue, causing the whole cell to shorten and bulge.
How is contraction triggered in smooth muscle? It seems more varied than in striated muscle.
It is much more varied. Smooth muscle contraction can be initiated by several types of stimuli. Mechanical impulses, like passive stretching of the muscle wall in the gut or blood vessels. Electrical depolarization, often triggered by neurotransmitters released from autonomic nerves. And chemical stimuli, like hormones, think oxytocin acting on the uterus, signaling molecules or changes in the local environment. But ultimately, all these diverse signals converge on one common pathway, increasing the concentration of intracellular calcium ions.
Okay, calcium is still key. But how does the calcium get into the cytoplasm if there are no T-tubules?
Good question. Smooth muscle cells lack the organized T-tubule system. Instead, they have numerous small invaginations of the cell membrane called caveolae. These caveolae, along with some associated vesicles and a less developed sarcoplasmic reticulum, are involved in controlling calcium entry from outside the cell and release from internal stores. Calcium delivery is generally slower and less explosive than in striated muscle.
And since there's no troponin, how does calcium actually switch on contraction?
It's a completely different mechanism, mediated by calmodulin and myosin light chain kinase, MLCK, figure 11.28. When intracellular calcium levels rise, calcium binds to a protein called calmodulin. This calcium-calmodulin complex then binds to and activates an enzyme, myosin light chain kinase, MLCK. Activated MLCK then phosphorylates as a phosphate group to one of the small protein subunits on the myosin head called the regulatory light chain.
And that phosphorylation is the switch.
That's the switch. Phosphorylation of the myosin light chain allows the myosin head to interact with actin and undergo the cross-bridge cycling, leading to contraction. It's a slower, enzyme-mediated process compared to the direct calcium-troponin interaction in striated muscle.
This sounds like it allows for more sustained contraction.
Exactly. And smooth muscle has another trick called the latch state. Once contraction is initiated, smooth muscle myosin can remain attached to actin for prolonged periods, maintaining tension or latch with very little ATP consumption.
Wow, super efficient for things like maintaining blood vessel tone.
Precisely. It allows smooth muscle to maintain tone or steady contractions for long durations without fatiguing easily or burning through lots of energy, essential for organs like blood vessels, airways, and the digestive tract.
So functionally, smooth muscle is geared towards slow, sustained, often involuntary contractions.
Yes. Think of the slow, wave-like peristaltic movements that propel food through your digestive system, the gradual constriction or dilation of blood vessels to regulate blood pressure, or the powerful, sustained contractions of the uterus during labor. It's often regulated by the autonomic nervous system, hormones, and local factors. The nerve endings often release neurotransmitters from swellings called boutons en passant that diffuse over a wider area to influence multiple cells rather than the precise one-to-one neuromuscular junctions of skeletal muscle.
And smooth muscle cells do more than just contract.
They do. They are actually quite active synthetically. They can synthesize and secrete components of the extracellular matrix, like collagen, elastin, and proteoglycans, contributing significantly to the structure of the tissues they inhabit, like blood vessel walls.
What about repair and renewal? How does smooth muscle compare here?
This is where smooth muscle really shines compared to striated muscle. Smooth muscle cells retain the ability to divide by mitosis throughout life. This means they can readily proliferate to increase their numbers when needed. For example, the massive growth of the uterine smooth muscle during pregnancy, or in response to injury, like in blood vessel walls.
So they can regenerate quite well.
Yes, they have a remarkable capacity for renewal and repair. They can also arise from the differentiation of other cells, like parasites associated with blood capillaries or even certain types of stem cells, much more adaptable than skeletal or cardiac muscle in this regard.
Okay, wow. We've covered skeletal, cardiac, and smooth muscle in detail. Let's try and pull this all together. Having explored the unique features of each, what's the big takeaway message here?
Well, if you connect this back to the bigger picture, what's truly remarkable is how these three muscle types, while clearly distinct in structure and function, all fundamentally rely on the interaction between actin or myosin to generate force. Yet through variations in cellular architecture like multinucleation versus single central nuclei, the presence or absence of striations and T-tubules, the type of cell-to-cell junctions like intercalated discs or gap junctions, and through vastly different regulatory mechanisms, troponin versus calmodulin MLCK, direct nerve control versus autonomic hormonal influence, they achieve incredibly diverse physiological roles. You can sort of summarize the key differences, right? You've got skeletal muscle, multinucleated, peripherally located nuclei, highly ordered sarcomeres causing striations, T-tubules forming triads, troponin-based calcium regulation, voluntary control via precise neuromuscular junctions, limited regeneration. Then cardiac muscle, typically single central nucleus, branched cells linked by intercalated discs, gap junctions, functional syncytium, striated with sarcomeres, T-tubules forming dyads, troponin regulation but triggered by calcium-induced calcium release, involuntary rhythmic contraction modulated by ANS, very poor regeneration, and finally smooth muscle. Single central nucleus, spindle-shaped cells often linked by gap junctions, no striations, different filament organization, no T-tubules, caveoli instead, colmodulin, MLCK-based calcium regulation, no troponin, capable of sustained latch state contractions, involuntary control by Anschromon's local factors, excellent regenerative capacity.
So these subtle microscopic differences, they translate into this enormous functional diversity we see in our bodies. It allows us to do everything from running a race to having our heart beat consistently for decades to digesting our food without even thinking about it. It really is just a marvel of biological engineering, isn't it? That really was a deep dive into muscle tissue histology. We hope you, listening in, now feel much more informed about these incredible and diverse tissues that power our lives.
Yeah, I think understanding these microscopic details, seeing how structure dictates function right down to the molecular level, gives you a whole new appreciation for the complexity and just the sheer elegance of the human body.
So maybe take a moment and think, what stands out to you from today's discussion? Considering conditions like muscular dystrophy or the impact of a heart attack really underscores how critical these cellular structures and molecular interactions truly are. It puts the unseen world inside us into perspective, doesn't it? Thank you for joining us on the deep dive. We hope you'll join us again next time for another journey into being well informed.